Good morning to you all. This is Raju, Department of Microbiology, Samhita Degree College. Today I am going to discuss on the topic called transposons. So in this topic we will discuss what is the definition of transposon and what are the transposon elements present in the prokaryotic organisms and how many types of transposons are there in the nature will discuss clearly. So first I'll tell you what is meant by transposon, right? Okay, so transposon can be defined as it is a DNA sequence that is able to move or insert itself at a new location in the same chromosome is called as a transposons, right? So primarily these transposons were first discovered by the scientist name called Barbara McClintock. The scientist name is called as Barbara McClintock in maize plants in the year 1940. So these transposons are also called as moving elements or jumping elements or controlling elements, right? So actually in prokaryotic organisms, if you see, we have majorly two types of transposable elements. So then tell me, what is the use of transposable elements? So actually these transposable elements in the nature which creates mutations to the DNA molecule, right? So these transposable elements majorly present in both eukaryotic and prokaryotic genetic materials. Okay, right. So in prokaryotic organism, if you see, we can find majorly two types of transposable elements. In that, number one, we can call it as a insertion sequences, or those are also called as IS elements. What is that? IS elements. Okay, so what is meant by insertion sequences or IS elements? Actually, they can be defined as these are the transposable sequences which can insert at different sites in the bacterial chromosome are called as insertion elements or insertion sequences. Actually, in IR elements which contains ITR structures in their formation. So IS elements which contains ITRs in their structures elements. So what is meant by ITRs? So ITR nothing but inverted terminal repeats. Inverted terminal repeats. So these inverted terminal repeats are majorly present in IS elements. IS elements. So these inverted or these ITR elements were first observed in E. coli. First observed in E. coli. If you see, the IS elements are relatively very short sequences containing molecules which having below 2500 BP pairs length. That means base pairs length. Okay. So the ITRs present at the end of ICS elements are important feature which enables their mobility. That means the ITRs which are present at the ends of the IS elements. So these are involved. These are useful for their mobility. అంటే ఏంటంటే ఈ ఐఎస్ ఎలిమెంట్స్లో మనకి ఎండింగ్ పొజిషన్లో ఇన్వర్టెడ్ రిపీట్స్ అనేవి ఉంటాయి. వాటిని ఐటీఆర్స్ అని చెప్పుకుంటాం. మరి ఐటీఆర్స్ యొక్క మెయిన్ ఫంక్షన్ ఏంటంటే దే ఆర్ మేజర్లీ యూజ్డ్ ఇన్ ది మొబిలిటీ ఆఫ్ ఐఎస్ ఎలిమెంట్స్ ఇన్ ద జీనోమిక్ మెటీరియల్, రైట్? ఓకే, యాక్చువల్లీ సో the length of the ITRs present in the IS elements of E. coli usually ranges from between 18 to 40 bps. 18 to 40 bps, right? So the best example for we can find number of IS elements in the nature. If if you see, there are number of several copies of IS elements like IS1, IS2 and IS3, IS4 and IS5, like different categories of IS elements we can observe in a bacterial chromosome, right? These are the types of IS elements present in the bacterial chromosome. Okay, and the second type of transposable elements present in the prokaryotic organisms are nothing but prokaryotic transposable elements. Actually, so what are these elements? What is the structure of these elements? We can discuss now. So prokaryotic transposon elements are also called as composite transposons. These are also called as composite transposons, and they are shown by a symbol called capital Tn. They are called a symbol called capital Tn. So composite transposons are also designated by the symbol called capital T small n. Actually, these type of transposons are made up of two types of IS sequences, which contains IS sequences. So one present at each end of the DNA sequence, which contains genes whose functions are not related to the transposition process, right? Okay, so these transposons have been found to have inverted repeats at the ends. Okay, so the length of these inverted repeats ranges from a few nucleotides about 1500 bps. Means, so actually ఇక్కడ చూస్తే వీటి యొక్క మనకి ఇన్వర్టెడ్ రిపీట్స్ అనేవి కూడా మనకి ఇందులో కనిపిస్తాయి. సో ఇందులో వీటి యొక్క లెంత్ చూస్తే మనకు సుమారుగా ఎంత ఉంటుందంటే 1500 బేస్ పేర్స్ లెంత్ అనేది మనకు కనబడుతుంది. మరి జనరల్గా సో ఎక్కడ మనకి ఈ టైప్ ఆఫ్ ట్రాన్స్పోజబుల్ ఎలిమెంట్స్ అనేవి కనిపిస్తూ ఉంటాయంటే, సో మేజర్లీ దే ఆర్ ప్రజెంట్. ద బెస్ట్ ఎగ్జాంపుల్ ఫర్ దీస్ టైప్ ఆఫ్ transposons, which includes the members of Tn1, Tn5, Tn9 and Tn10 are the best example for this type of composite transposons. So actually in nature we have different types of transposons based upon the transposition mechanism. Okay, actually we have to know what is meant by transposition mechanism. So transposition mechanism is nothing but transfer of one transposon from one place and attached into the another place of a same DNA is called as the transposition mechanism. So based upon the transposition mechanism, these transposons are mainly classified into three categories. These transposons are mainly classified into three categories. So what are those three categories of transposons? In that, first one is called as cut and paste model of transposons. The first one is called as cut and paste model of transposons, and second one is called as replicative transposons. The second one is called as replicative transposons, and third one is called as retro elements or retro transposons. So these are the three types of transposon based upon their transition mechanism. Now individually we discuss a transposon mechanism of cut and paste, replicative and retro transposons. So this is one of the three types of transposons, that is called as cut and paste mode of transposons. So in this type of mechanism, how transposons will be jump from one place to another place, we can see clearly, right? So జాగ్రత్తగా వినంటే మీకు ఈజీగా అర్థమవుతుంది. Actually this is the DNA structure of a donor DNA. సో డోనర్ డిఎన్ఏ అంటే ఏంటంటే ఇక్కడ మనకి ఎక్కడి నుంచి అయితే ట్రాన్స్పోజన్స్ అనేవి కట్ అవుతాయో అక్కడ కట్ అయ్యేటువంటి డిఎన్ఏని మనం ఏమంటామండి డోనర్ డిఎన్ఏ అని చెప్పుకుంటాం, రైట్. ఓకే, మరి ఎక్కడి నుంచి కట్ అయినటువంటివి ట్రాన్స్పోజన్స్ వెళ్ళి అటాచ్ అవుతాయో సో దాన్ని మనం ఏంటంటే రెసిపియెంట్ డిఎన్ఏ అని చెప్తాం, రైట్. ఓకే, సో మరి ఇక్కడ మెకానిజం చూస్తే మనకు ఎలా జరుగుతుందంటే సో ఇన్ కట్ అండ్ పేస్ట్ టైప్ ఆఫ్ ట్రాన్స్పోజన్స్ ఫస్ట్ an enzyme called for this mechanism there is an involvement of an enzyme called as transposase. The enzyme called is transposase. This transposase enzyme which involved in the cutting and pasting of the transposons present in the DNA structure, right. Okay, so actually in cut and paste type of transposon what will happen, see clearly. So this is the donor structure of DNA. So in the donor structure the striped area is called as transposons material containing area. So this total area called as transposons containing area. So actually when there is a transposition mechanism occur, this transposase enzyme come and attach to the ends of the transposon materials, come and attach to the ends of the transposon material in the DNA structure, right. Okay, then what will happen here means this transposase enzyme cuts the transposon sequence from the donor sequence and separated like this. So these are the excision parts of the transposons. అంటే ఏంటి, ఇక్కడ మనకి ట్రాన్స్పోజిషన్ ఎంజైమ్ వచ్చి ఈ ఎండింగ్ పొజిషన్ దగ్గర మనకి ఏర్పాటు చేసుకుని అక్కడ ఎండింగ్ పొజిషన్ దగ్గర మన కటింగ్ అనేది జరుగుతుంది. సో ఎప్పుడైతే కట్ అయిందో ఇక్కడ నుంచి మనకి ఈ ట్రాన్స్పోజబుల్ ఎలిమెంట్స్ అనేవి కట్ అయిపోయి డోనర్ డిఎన్ఏ నుంచి మనకి ఇలా సపరేట్ అవడం అని జరుగుతుంది. సో వీటిని మనం ఏమంటామంటే దీస్ ఆర్ ది ఎగ్జిషన్ పార్ట్స్ ఆఫ్ ది ట్రాన్స్పోజన్స్ అని చెప్పుకుంటాం. అంటే ఏంటి, ఇవి కట్ అయినటువంటి ట్రాన్స్పోజన్స్ మెటీరియల్. దెన్ these excited parts of transposons will transfer by transposase to the another part of the same genomic material. That part is called as recipient part of the DNA. అంటే ఇక్కడ ఏంటి recipient part of the DNA అంటే మీనింగ్ ఏంటి అంటే సో ఇది కూడా సేమ్ DNA లో స్ట్రక్చరే, బట్ ఇక్కడ ప్లేస్ 1 నుంచి ప్లేస్ 2కి ఈ ట్రాన్స్పోజబుల్ ఎలిమెంట్స్ అనేవి జంప్ అవుతూ ఉంటాయి అన్నమాట. మరి ఇక్కడ చూస్తే ఇక్కడ కట్ అయినటువంటి పీసెస్ ఏవైతే ఉన్నాయో ఈ పీసెస్ ని ఈ ట్రాన్స్పోజేజ్ అనే ఎంజైమ్ వేరే లొకేషన్ లోకి తీసుకెళ్లి అక్కడ మనకి అటాచ్ చేయడం అనేది జరుగుతుంది. సో దట్ పార్ట్ ఈస్ కాల్డ్ యాజ్ recipient site or recipient site of DNA. So here finally there is a attachment of excision transposons in the recipient DNA. Now final products if you see, so this is the recipient DNA with the transposons and this is the donor part of the DNA. If you see the structure and length of these two types of recipient DNA with transposons and donor DNA with transposons, there is a variation in the size. Why this variation they called it means actually సో ఇక్కడ మనకి డిఎన్ఏ ఏదైతే డోనర్ డిఎన్ఏ ఉందో డోనర్ డిఎన్ఏలో మనం ఒక పీసు ట్రాన్స్పోజన్ పీస్ కట్ అవ్వడం వల్ల ఏమవుతుందంటే ఇక్కడ లెంత్ అనేది మనకి తగ్గిపోతుంది. బట్ ఆల్రెడీ ఇక్కడ మనకి రిసిపియెంట్ డిఎన్ఏలో ఎక్స్ట్రా ట్రాన్స్పోజన్ ఉంటుంది కాబట్టి దానికి ఈ పీసెడ్లు అటాచ్ అవ్వడం వల్ల ఏమవుతుందంటే దాని యొక్క లెంగ్త్ అనేది ఇంక్రీజ్ అవుతుంది. ఎక్కడంటే there is an increase in the length of recipient DNA, so that after attachment of this excised transposon from donor DNA to recipient DNA, in this case there is a increase in the length of the recipient DNA with transposon, but there is a decrease in the length of the donor DNA of transposon is occurs. So this type of transposons are called as cut and paste. Why these are called as cut and paste means here the transposase enzyme cutting the transposon material and it is attaching or pasting and some other place, so then it is called as cut and paste type of transposons, right? So this is the one of the method, one of the category and one of the type of transposon. Next we will go for second mode of transposons mechanism.